Today we're going to look at Module 11, Plant Diversity and Reproduction. Let's not forget what a basic plant cell looks like. These cells have chloroplasts, central vacuole, cell walls, a lot of structures we don't see in animal cells. When we're talking about plants, we can talk about the study of plants. That's botany. If you study plants, you're a botanist. And we see that all plants have this dual life cycle called alternation of generations, where there exists in the life cycle both a multicellular diploid form and a multicellular haploid form. Let's take a step back and remember that we're working our way through the domains of living things. We've already gone through bacteria and archaea, and we've looked at in eukarya algae, mushrooms, and now we're looking at plants. So we looked at protists and fungi, and we're also eventually, after plants, going to make our way to kingdom Animalia. But we're still very staunchly in domain eukarya. The alternation of generation life cycle contains two multicellular stages, one the gametophyte and one the sporophyte. The gametophyte produces gametes. Those gametes fuse to form a zygote, and that zygote can develop into the diploid sporophyte. The gametophyte is haploid, which makes sense because gametes are haploid or have half the genetic material of the diploid organism, and sporophytes are going to be diploid. And the sporophyte multicellular stage produces spores. Let's begin with the non-vascular plants like mosses, hornworts, and liverworts. The non-vascular plants do not have vascular tissue. Vascular tissue is a system of tube-shaped cells branching through a plant that transports materials between roots and shoots. The bryophytes, or the non-vascular plants, do not have this tissue, whereas the tracheophytes, or the vascular plants, will. When we're looking at the bryophytes, we're looking at organisms that grow in forests, where the soil is cool and moist. They're low growing because they don't have that structure, that vascular tissue that could allow them to grow very tall. They actually have to rely on osmosis, that passive process that draws water into their tissues, to their cells. So that's why they need to grow in a cool and moist environment. They have shoots and rhizoids. The leafy shoots are stem-like. The rhizoids are kind of root-like, but rhizoids are actually long, thin cells, and they're used mainly to anchor plants like moss. And they can only transport water and other nutrients by osmosis, so they're not true roots. The three main types of bryophyta are moss, liverworts, and hornworts. They all last vascular tissue, hence being in the bryophyta class. Their gametophyte or haploid stage is actually their dominant phase. That's the dominant phase in their life cycle. Other plants would actually have the sporophyte as their dominant phase. That sets bryophyta apart. If you're looking at mosses, you would notice that they're small and flowerless. They usually grow in clumps or maybe a mat, but they're going to be in damp, shady locations. They are generally composed of leaves that are one cell thick, covering a very thin central stem. But this central stem would not conduct water. Remember, it's non-vascular. Mosses do not have seeds, as well as being absent in vascular tissue. Liverworts look like flat leaves. They tend to have a lobed appearance, and then you can see the sporophyte here has that umbrella shape. They absorb water directly through their leaf-like structures, so they do have to live year-round as well in a moist soil. Now they're actually named after the shapes of their flat leaf-like structures because those lobes look like the lobes of a liver. Now when the gametophyte matures, it produces structures that resemble tiny green umbrellas, inside of which egg and sperm are produced. So these are actually where we're going to see the haploid cells called gametes produced. So that is the gametophyte down at the bottom and the sporophyte up top. And we see a similar structure with the hornworts. They, instead of having the umbrella, have just these projections. And in their gametophyte stage, they have these broad, thin, leafy structures that absorb water directly from the surface of the soil produced. Looking at the reproductive life cycle of a bryophyte shouldn't be new. It's still alternation of generations with multicellular diploid and haploid states. with the gametophyte generation producing male and female gametes that develop structures that eventually produce sperm and egg, and those would be fertilized in order to produce the sporophyte. Now that's pretty much it for the non-vascular plants. We're not going to go into much more detail. But let's move into the vascular plants. And we'll begin with the seedless vascular plants. But before we do so, let's establish what is that vascular tissue that was missing in the bryophytes. Well, vascular tissue of plants is a complex conducting network of tissue. The two main components are xylem and phloem. Now, xylem consists of dead cells, and phloem consists of living cells. And the xylem mainly conducts or transports water and stuff dissolved in that water, so water and small nutrients. Whereas phloem, those living cells, they distribute mainly larger nutrients like sugars and amino acids throughout the plant. You can see here in the cross section of this piece of celery some of the vascular bundles. The cells of xylem and phloem are typically long and slender. They are there to conduct water or nutrients, including things like minerals and ions, through the plant. They're similar to pipes, but they're going to use the hydrogen component of water in order to allow it to move nutrients and other substances throughout the plant. What I mean by the hydrogen component of water is the use of hydrogen binding to other atoms in separate molecules can create hydrogen bonds. Hydrogen bonding is responsible for cohesion and adhesion that we see in xylem and phloem that make these tube-like structures good conducting material. So here you can see the basic tube-like structures of the xylem and phloem. The xylem is in pink and the phloem is in blue-green. We can call it blue-green. And the pink xylem consists of dead cells, so note they are thinner and flatter. And then the phloem consists of living cells. All around it are mesophyll cells and parenchyma, which are structural support and are the cells that perform photosynthesis in a plant. Here, looking at some of the vascular tissue for plants, we can see structures like the pith, which is soft, spongy central tissue, but it's not germinal or vascular. And then we can see at 3 and 4, the xylem and phloem. So it is arranged in a particular location and area depending on the type of plant. Vascular tissue is designed for height. Ferns, pteridophytes, other organisms that have vascular tissue within them, they are going to be much taller than the bryophytes. There's a compound called lignin, and it's a polymer found in the cell walls of plant cells, and it makes up the vascular tissue. It makes those tube-like structures rigid. The lignin hardened vascular tissue is specialized to conduct water and to transport sugar. And remember, sugar is the product of photosynthesis. So it makes sense that these tube-like structures would be in organisms that primarily perform photosynthesis over those that do not. And lignin is incredibly strong. It can support not only ferns and other vascular plants, it can support things like trees. Let's look at the major group of seedless vascular plants, and that is mainly going to be ferns. But these are the major groups. Xylophyta, the whisk ferns, Sphenophyta, the horsetails, Lycophyta, the club mosses, and Pterophyta, just the ferns. They all reproduce via spores. They have no seeds, no flowers. And there are various important parts of its life cycle, like the use of the multicellular gametophyte and sporophyte phases, because ferns are also going to reproduce using alternation of generations. And we'll look briefly at the sori and spore production. A fern sporophyte is what you would recognize as a fern, and it would produce spores on the back of its blades. The location of where these spores are held are these tiny clusters called sori. A sorus is a cluster of sporangia or spore producing tissue, and the spores of ferns are actually going to be utilized for asexual reproduction and are usually dispersed via the wind. Now, in unfavorable conditions, the spores are the products of meiosis in the sporophytes, and in that case, we may see the production of the gametophyte multicellular individual. The fern gametophyte is going to be something you don't recognize as much. It's haploid, it's multicellular, and it developed from mitotic cell division. After release of spores, they would multiply to create this multicellular organism. The gametophytes essentially come in two flavors, the hermaphroditic, or male and female version that exist in the same gametophyte, or just the male gametophyte. So here you can see the life cycle of the fern, and it consists also of a multicellular gametophyte and sporophyte generation. You see the back and forth, and this is also true alternation of generations. We see that the sporophyte generation is the one that would produce the spores, and the gametophyte generation is the one that would produce the gametes. Horsetails and club mosses are interesting because their cells contain crystals of silica that's similar to the base skeleton of diatoms, a type of protist. And the leaves of horsetails and club mosses are going to be arranged in circles around a central stem, and that's what gives the appearance of a horse's tail, and it's hence where they have garnered their name. Club mosses are a little bit smaller than horsetails. They're small plants that look a lot like miniature pine trees, and they're sometimes referred to as ground pines. And so that's it for the seedless vascular plants. They're a little bit taller, but they're not a tree or a flowering plant. So let's look at some of those examples, like the gymnosperms, the cycads, the ginkgos, the pines and spruces, and Cycadophyta, Ginkgophyta, and Coniferophyta, respectively. Now, gymnosperm as a word comes from gymnos and sperma, meaning naked seed. And that's talking about the unenclosed condition of the seeds in the gymnosperms. That naked seed condition contrasts very much with the seeds and ovules of the angiosperms, which we'll look at next. These flowering plants would encase or enclose their seed or ovary within a protective structure. Now, pine seeds, being in gymnosperms, develop either on the surface of scales or leaves, often modified to form cones, but they are not 100% protected and enclosed. These seed plants can actually live on almost any environment, from moist habitats to cold and dry habitats. Let's look here at the male and female pine cone. The male pine cone is called the microstrobili, and the female pine cone is called the megastrobillus. And the female pine cone contains ovules that can be fertilized by pollen, and that would make them become seeds. And the male pine cone contains areas called microsporophylls, which are modified leaves, and they extend out from a central axis, but within each microsporophyll would be many microsporangia or pollen sacs. Pine pollen is going to contain sperm. A lot of modern conifers, including pines, have to be wind pollinated. Now, the cycads are a little bit more insect pollinated than wind pollinated, but wind pollination is still going to abound for the gymnosperms. And pollen, again, that fine dust that contains gametes, is containing the sperm for the seed producing plant. Let's look at the seed of a gymnosperm. So we have the plant embryo inside, it's packaged with a food supply, and it generally has a hard protective seed coat. Gymnosperms produce and bear their seeds directly on seed cones, not enclosed, hence gymnosperm meaning naked seed, and that is different than the angiosperms that protect their seeds inside a layer of tissue. The embryo for a gymnosperm is a tiny sporophyte formed after fertilization in a seed cone. And the food supply will provide the growing embryo with nutrients until the seed can actually be transported to a location where it can germinate. And then it would surround and protect itself until we got sprouts or a shoot and root system. The reproductive life cycle for gymnosperms is also going to be alternation of generations, where we see both a haploid and diploid multicellular life stage, with the haploid stage being the gametophyte and the diploid stage being the sporophyte. Next, we're going to look at the angiosperms. And angiosperms are flowering plants, and they're going to protect their fertilized seed. Now, angiosperms are not always going to be the flowering plants that you associate with bright colors. For example, we can see that the tassels on the top of this corn plant, those would have to be the flowers. And because of the differing varieties that we have when it comes to angiosperms, there's some classifications we can apply. Like, is the angiosperm a monocot or a dicot? Woody or herbaceous, or a biennial or perennial? Monocots and dicots are primarily looking at the number of seed leaves that are in the seed itself. If you have 2 seed leaves, or cotyledons, then it is a dicot. If you have 1, it's a monocot. Something like a bean would be an example of a dicot. Something like a corn seed, that'd be an example of a monocot. But there are things besides just single versus double cotyledons that would tell if something is a monocot or dicot. You don't always have access to the germinating bean or seed. So you could look at the leaf. If it's a long, narrow leaf with parallel veins, it's a monocot. It's a broadleaf that has a network of veins. It's a dicot. If the vascular bundles are scattered and not arranged, then monocot. If the vascular bundles are in a ring, then dicot. If the floral parts are in multiples of 3, monocot. And if the floral parts are in multiples of 4 and 5, dicot. So here we can see a couple of different examples. We have a monocot here with the parallel leaves. We can see some netting in many of the others, making that a dicot. Corn, with its flower right here, would actually be a monocot. And these would be monocots as well, even though the leaves radiate around a central axis. Let's talk about woody versus herbaceous. Woody or herbaceous plants are both considered angiosperms. The cells of woody plants have thick cell walls and they give more support to the plant. Something like a tree and a shrub, rhododendrons, roses, they'd all be examples of woody plants. Now, vines, ivy, grapevines, also woody plants. An herbaceous plant would not have a woody stem. Instead, their stem is smooth and flexible. An herbaceous plant example would be something like a dandelion, a sunflower, a zinnia. Perennial plants grow year after year. Annual plants are plants that live for only 1 year, and biennial plants are plants that live for 2 years. And generally, they have 2 seasons of growth, one with food storage and one that produces flowers and focuses on reproduction. And those would be the differences between annuals, biennials, and perennial angiosperms. Just a further classification we could look at. Let's also look at the anatomy of a flower. And this is a generalization, but there's 5 basic parts to a flower: the pedicel, the sepals, the petals, the carpel, and the stamen. The carpel is the female part of the flower, the stamen is the male part of the flower, and they're geared towards reproduction. The petals are leaf-like, but they're above the sepals, and the sepals are generally green or less colorful than the petals, and these are structures above the pedicel that protect the petals. And the pedicel is the stem that holds the flowers. Here we can see some of those structures. We can see number 1, the sepal, is below number 2, the petal. We can also see that stigma, style, and ovary are the 3 parts of the female reproductive structure. Number 4, the carpel. And we can see the stamen, which is in yellow, labeled number 3. The stamen actually consists of 2 parts: the anther, which contains the pollen grains, and the filament. So we can look at it over here as well. The anther and the filament collectively are called the stamen. The sticky bulb here is the stigma, The area where we'd have a pollen tube is the style, and the area with many ovules is the ovary. We can also see the petals a little bit better, and we can see some sepals. And then down here is that pedicel. So that's the base anatomy for a flowering plant. Now keep in mind that the stamens that have long filaments are essentially serving as stalks that support the anther, holding those pollen grains in place. The pollen grains contain haploid gametes, and they would essentially be the sperm of the plant. And they're hanging out near the stigma, or the covered, sticky substance covered part of the carpel. Carpel, the female reproductive structure, is made up of the stigma, style, and ovary. And that sticky substance on the stigma is designed to catch pollen grains. The style is an extension of the ovary, generally contains the pollen tube. It's designed to hold the stigma up so it can be exposed to the pollen grains. And the ovary contains many ovules, and that's where the embryo sac would develop. And that embryo sac would contain the eggs of the plant, and that would be where the seed develops if fertilization occurs. So again, carpel, it's the female reproductive structure made up of the stigma, style, and ovary. The stamen is the male reproductive structure, and it's made up of the anther and filament. Now, there are exceptions. Not all flowers have the standard 5 parts, and there's exceptions to a lot of things in biology. Now, flowers that have both the male and female parts of the flower, they're considered perfect flowers. Others that don't have both, and they're considered imperfect. In some plants, the male and female flower parts, they mature at different times. And while most plants self-fertilize, not all could be, depending on these variations. Some plants have chemical mechanisms that reject their own pollen. Other plants will happily accept pollen from another plant of the same type, but rejects its own. Some plants have imperfect flowers, so each specimen is only male or female and does not contain reproductive structures. All of these would prevent plant self-fertilization. Let's look at the petals as well. Not only are they colorful, they generally serve a purpose, such as attracting pollinators, like birds and insects, to the flower. Pollination can also be accomplished by wind, but oftentimes other organisms can aid the process of fertilization. Now, not always do we have very colorful flowers. Other times they're emitting a smell. So smell and color can be attractants to pollinators. But others that are not colorful or don't smell a certain way might just be wind pollinated, like the cornflower here. Now, insect pollinated flowers do not have to smell good. They don't have to be pleasant, like the aroma of a rose. They could smell like rotting flesh. That could attract flies, for example, that then can pollinate other similar plants they're attracted to. When you look at flower taxonomy, again, the biggest thing is the monocot dicot structure. And we know monocots have flower parts in multiples of 3, while dicots have flower parts in multiples of 4 or 5. But that means that dicot flowers have either 4 or 5 petals and 4 or 5 stamens, whereas the typical monocot has either 3, 6, or 9 petals, and usually 3 or 6. So it's not looking for just 4 or 5, or just 3. Multiples of each also apply. We can also look at composite versus simple flowers, and a simple flower is made up of just one flower, like the tulip. A composite flower is made up of several smaller flowers. For example, the sunflower. When we're looking at the reproduction of angiosperms, we have to acknowledge that alternation of generation is still the main process. But looking at the flower structure, we can also see that the reproductive structures have been brought together in a lot of complete flowers, or perfect flowers. And the stamens would produce the pollen grains in that case, which contains sperm. And the pollen grains should be able to make their way to the embryo sac, which are in the ovary of the carpel. That's where the eggs would be. And the process of pollen grain formation is going to be quite different from the process of egg formation. Each anther is actually going to be a flower's stamen that has regions called pollen sacs. And what that means is that on the tip of a stamen, where the anther is located, are areas that contain pollen grains. The spaces that contain the pollen grains are called pollen sacs. In the pollen sacs, the diploid cells have undergone meiosis in order to form pollen that is haploid. Once that meiosis occurs, there will be at least 4 haploid cells produced from the one round of meiosis. They are generally referred to as microspores. Microspores have to be encased in thick walls to protect them, and it's to protect them from unfavorable conditions. But while encased, it can undergo mitosis and basically copy itself and make more pollen. That results in a lot of haploid cells inside the anther casing, and so we see many rounds of mitosis producing a lot of pollen. And once there are at least 2 cells inside the casing, one of the cells differentiates and becomes a tube nucleus, and the other cell differentiates into a sperm cell. These cells, encased in a protective coating, they make up the pollen grain. And each pollen sac in a mature anther contains hundreds or even thousands of pollen grains. So to simplify that, the pollen grains are in the anther of the flower's stamen. In order to create haploid pollen, we have meiosis, but mitosis follows that process, turning microspores into double nucleide structures, one of which will help produce the pollen tube. So here you can see the base location for pollen grains in the anther. You can see that microspores are created, but then quickly differentiate into something that has 2 nuclei, and that's going to be something that allows for the process of fertilization via the use or utilization of the carpel. Here's what angiosperm pollen looks like up close on the head of a pin on the right, or under a scanning electron micrograph on the left. Let's also now talk about egg cell formation. So on the inside wall of the ovary, there's a swelling, and if it begins to grow, that's the beginning of a new ovule. When the ovule grows, a single diploid cell undergoes meiosis, resulting in 4 haploid cells. In this case of animal female meiosis, only 3 of those haploid cells are destroyed. They're considered useless and they disintegrate. And there's only one remaining cell that gets called the megaspore that can undergo forms of mitosis to essentially form multiple nuclei. Now, the cell's undergoing mitosis multiple times, so it's getting bigger, but the cell never divides. So it creates a multinucleated megaspore. It'll actually have 8 haploid nuclei. The cell walls and membranes that form around this megaspore help support it and keep it together. And that actually results in a large central cell with 2 nuclei and 6 smaller cells, 3 of which are on one side of the larger cell and 3 of which are on the other side. And this arrangement of 7 cells is actually called the embryo sac. So the formation of the embryo sac is going to rely on meiosis followed by mitosis, but no division. And this creates a megaspore, which essentially is a multinucleated cell, and those nuclei will separate or spread out in order to complete the formation of the egg sac. Let's overview. Since the pollen grains contain at least 2 cells, and since the embryo sac contains 7 cells, both are multicellular structures. They both produce gametes, so you can interpret this as an alternation of generation life cycle. And if you look at the angiosperm life cycle in this way, the plant itself is the sporophyte, which through meiosis produces the pollen grains, and the embryo sacs, which form the gametophyte generation. Once the 2 gametes participate in fertilization, a diploid zygote results, which gives rise to the sporophyte generation. So keep in mind that even angiosperms use alternation of generation, but they use mitosis in order to create varied types of gametes that then come together via fertilization to create the diploid organism that we tend to call the sporophyte. Reproduction in angiosperms definitely relies on pollination as well. or the transfer of pollen grains from the anther to the carpel in flowering plants. While wind pollination is possible, insects and birds are going to be primary pollinators for many flowers. Birds and insects also benefit. They eat the nectar for food, the nectar that is inside plants. But while they're doing so, pollen grains get stuck on them, and then they travel from flower to flower and brush up against the carpel of other flowers, and the pollen will leave the insects or birds and sticks to the stigma of the carpel. And that would complete the pollination process by birds or insects. In order for the sperm to get to the egg, the sperm of the pollen grain has to travel down the carpel to the ovule. And that means when the pollen lands on the stigma, the tube nucleus begins to undergo mitosis and differentiation. This results in a tube called a pollen tube. So the second nucleus of the developing sperm has to be used to create a pollen tube. That allows the other pollen nuclei to get to the embryo sac. And if the pollen tube contained only one sperm cell, mitosis would still occur in the pollen tube so that there are now two sperm cells. But either way, two sperm cells make their way to the ovule via the pollen tube. Once the two sperm cells have reached the embryo sac, a rather unusual thing happens. One of these sperm cells fuses with one of the 6 small cells in the egg of the embryo sac. When that type of fertilization occurs, a zygote is formed, and the other 5 small cells disintegrate. The second sperm cell travels to the large double nucleus cell in the middle of the ovule and fuses with its 2 nuclei. The cell that results, called the endosperm, therefore contains extra genetic information. Whereas the zygote begins to develop in the seedling of the plant, the endosperm begins to develop into a food source for that zygote. This type of fertilization is called double fertilization, and it's completely unique to Kingdom Plantae. Double fertilization is a fertilization process that requires 2 sperm to fuse with 2 other cells. The other cells are generally in the embryo sac. The endosperm that develops is the food source for the growing zygote that will become the embryo. And the cotyledons will form not too long after that. They'll be present in the developing plant, and they either absorb the remaining endosperm and then provide nutrition themselves, or they produce enzymes that help the embryo absorb more nutrients from their endosperm. But regardless, once an embryo fully forms its cotyledons, it's considered mature. So for a monocot, you have one cotyledon, a seed coat that protects the seed, and you should see after germination a plumule and radicle for the development of the shoot and root, and the endosperm would be the food supply for the seed. For a dicot, you should see 2 cotyledons, still a protective seed coat. There should be an area where the 2 sides attach. That's called the helix or hilum. And you should see an early shoot and root of the developing seed. And then near the end of seed development, the plant severs its connection to the ovule, and the ovule begins to develop a protective coating, and that's called a testa. Once the testa has been formed, the ovule is called a seed. Then the ovary begins to swell and mature, becoming the fruit of the plant. The seed would be just the ovule with a protective coating encased in a mature plant embryo and a nutrient source, whereas the fruit that develops around it is a mature ovary that contains a seed or seeds. There's different types of fruit, and we can look at simple fruits, and they can be fleshy or dry. When a fruit forms from a single ovary, that's when it's considered a simple fruit. Fleshy fruits are things like apples and pears. There's drupes like olives and peaches, or berries, which include tomatoes and grapes, or even modified berries, cucumbers, and oranges. They would all be considered fleshy fruits. Dry fruits could be pods, beans, peanuts, poppy seeds, maple tree fruits, walnuts, acorns, corn, sunflowers. And fruits that form from many ovaries are called compound fruits, like that of raspberries and strawberries, or pineapples, figs, and mulberries. Here are just some types. You can see that the samara of the maple tree, which you may have seen before, those are dry fruits, whereas something like an apple would be a fleshy fruit. And so there's just different categorizations for the base structure of fruit. When warm water surrounds the seed that is germinating, that's when the outer covering or the testa would loosen, and that would allow water to get inside the seed, and that would trigger growth processes that would finish out the germination process. So, germination and early growth are going to be triggered by a lot of different growth signals and chemicals, and it's when the plant breaks the soil that it's typically called a seedling. And at this point, you should have cotyledon or cotyledons begin to open, forming kind of the immediate leaf-like structures or early shoots and leaves. In a lot of plants, the cotyledons perform photosynthesis until leaves can form and take over. The true leaves of the plant do tend to grow eventually, and they will form from the top portion of the embryo, and that's called the epicotyl or early shoot. In some plants, the embryo forms the beginning of its true leaves in the seed. That occurs in a structure called a plumule. Let's look at some words here. So the radicle is going to be an early root. You can also call the hypocotyl an early root. The cotyledon is a young shoot. And the cotyledon leaves will do photosynthesis until the epicotyl can produce true foliage leaves. There's different types of reproduction for plants. It's not all alternation of generations. There's some asexual reproduction, and that's generally called vegetative reproduction. Like grass and many weeds reproduce asexually. Or we can see the propagation of potatoes from the formation of eyes, and in the planting of the eyes for more potato growth. The offspring of vegetative reproduction are genetically identical to the parent plant, hence it being true asexual reproduction. A lot of commercial gardeners would use these methods to clone certain plants. The resulting plant from vegetative reproduction is a clone. So here you can see bulbs and stolons, so horizontal roots or eyes from potatoes, or you could take a leaf cutting. All of those are methods of asexual reproduction. And that is our last topic for chapter 11. So make sure to review, and we will do chapter 12 next time.